Hi, this is Dr. Biology here, and this video is about how enzymes work. When you eat, your body goes straight into action to start to digest the food that you've eaten. It needs to make it into smaller, soluble molecules to be absorbed through the small intestine into the blood. To do this, as we know already, many processes occur, like mechanical and physical digestion, as well as things like hydrochloric acid in the stomach. But the most important process is when enzymes come into play. So, before we talk about digestive enzymes in my next video, we need to understand a bit about the nature of enzyme molecules and explain how they break down food. We need to before we do that, we need to learn what we call the lock and key theory, which is a simplified model to explain enzyme action. So, what are enzymes? Well, what does this picture show us? Well, it looks a bit like alphabetti spaghetti, but in fact this is a computer model of an actual enzyme. But it looks very complicated, so I've decided we're not going to use this, we're going to use this instead. So first of all, enzymes are proteins. It means that they're made up of long chains of amino acids. One key thing students get wrong is that they seem to think enzymes are alive or dead. Okay, they're not. They're just long strings of amino acids folded up in such a way as to make a three-dimensional shape. They have an active site that has a unique shape, so it can bind with a specific substrate molecule, such as carbohydrates, proteins and lipids. So why are they so important? Well, they do speed up or catalyze reactions, such as in digestion. If you didn't have any enzymes, digestive enzymes in your digestive system, it would take too long for the food to be broken down by other means, and therefore you wouldn't get enough energy from respiration and you would die. So let's look in your digestive system. Well, the first thing you would see is lots of substrates, and all these substrates are mixing up. Okay, this could be in your stomach or in your small intestine, and they're all moving around. And within there, you've also got your enzymes. Okay, and so these enzymes will be also moving around because they have kinetic energy. Okay, and they'll be moving around. And as they move around, some of them at some time or another may start to collide. So boom, this one collides with that one there. So the enzyme collides with the substrate. So if we just look at one enzyme and one substrate, you can see that the enzyme is like a lock. Okay, the enzyme has an active site with a specific shape, and the substrate has a shape that will fit into this active site, a bit like a key going into a lock. And this will form what we call an enzyme-substrate complex. This means that the enzyme will now break down the substrate into a small product or small products. So these products are going to be smaller usually. If we're talking about a digestive enzyme, they will break substrates into smaller products. Now, as I said before, enzymes are specific. So what I mean by specific, they're specific to one substrate. And in this example here, I've got another type of substrate, and as you can see, as it comes close to the active site, it's not a similar shape. It won't fit into that active site. So if it won't fit, then no reaction will take place. So enzymes are not changed themselves by the catalyzed reaction, and so the great thing for digestion is that they can continue to collide with more substrates and catalyze more reactions. This allows food molecules to be quickly broken down in the digestive system. So you will now need to know what enzymes are found where and how they break down different food molecules in the digestive system. That will be my next video, so I will see you soon, and I hope you found this useful.